Good afternoon and welcome to today's Connect Science seminar. It's my pleasure. This is David Fisher from MGH. It's my pleasure to welcome everybody and to take a few moments to introduce our speaker for this afternoon. Our speaker is Dr. Sean DeMary, who is in the dermatology department at Mass General. Sean is a rising superstar in the field of immunology at Harvard Medical School, and has already made some fantastic contributions, which I'll very briefly share with you. He was born in Tehran and immigrated to the United States, attended college at Washington State University, and was in the MD PhD program at WashU in St. Louis, where he did his PhD with Rafi Kopan. He was then a resident in dermatology at WashU at Barnes Jewish Hospital, and did a postdoc fellowship with Wayne Yokoyama, also in immunology. And then we were fortunate enough to recruit him here to Mass General in dermatology and also in the Cancer Center at MGH back in 2015. In 2018, he became the director of our high-risk skin cancer clinic, and more recently also the director of the Center for Squamous Cell Carcinoma of the Skin here at MGH. He's won numerous awards, and I do want to highlight just a couple of them because they're pretty cool. In 1998, he won the silver medal in National Chemistry Olympiad in Iran. And the following year, 1999, he won the gold medal in the National Biology Olympiad in Iran. So this is pretty remarkable stuff. Since he came here, he's won NIH Director's Early Independence Award, Young Physician Scientist Award from American Society of Clinical Investigation, and also recently a regular inductee into ASCI, American Society for Clinical Investigation. He recently won the Howard Goodman Award, which is a very prestigious research award given selected to a single individual in the entire research community of MGH that comes with a large financial award as well. His research has focused on several key aspects of immunology, and just two that I would very briefly highlight are studies that he carried out from the most basic science all the way to clinical trial and validation on the role of inducing the expression of TSLP, the lymphopoietin, thymic stromal lymphopoietin, and its activity in preventing skin cancer, skin carcinogenesis, and his discovery that calcipotriol can induce expression of this factor, and then testing and demonstrating that this can have an effect in skin cancer prevention, and is now already, this is a clinical strategy that's in wide common usage in numerous places around the world. He also published a lovely paper a couple of years ago in Nature describing immunity to commensal papillomaviruses as a means of protecting against skin cancer, and numerous other aspects of immunology largely related to cancer. His topic for this afternoon's lecture is NK cell function in peripheral tissues, and it's our pleasure to welcome Sean.
Thank you so much, David, for the very kind introduction, and I'm delighted to be presenting at this forum.
Sean, I apologize. There was one other thing I needed to say,
Absolutely.
Which is to announce that for anybody who has questions, the way we do this is we ask you to post them in the Q&A tab down at the bottom, but not in the chat. So we will read off the questions at the end. Feel free to type them in as the lecture is going along. And but again, Q&A, not into the chat. Okay, sorry about that.
Absolutely. Thank you again for the introduction, and thanks for this opportunity to present in this forum. As was mentioned, my lab studies cancer immunology and mostly what we call cancer immunoprevention. Today's topic will be focusing on one of our more recent discoveries in the realms of engaging and using NK cells as a means for regulating tissue immunology in peripheral tissues. But before I get into that, I do want to mention and a little bit elaborate on what my laboratory broadly is interested in, which is cancer immunoprevention. As everybody on this meeting and this call knows, that there has been a fantastic and massive amount of expansion on our understanding of how the immune system can be utilized and activated and potentiated against advanced disease, advanced cancer for patients with metastatic disease. However, the focus in our laboratory has been around the understanding of the role of the immune system in regulating how cancers form in peripheral tissues, namely in epithelial organs. And we really think of ourselves as epithelial immunologists that look at epithelial cells as the source of that recognition for immunity. In other words, as the epithelial cells undergo early stage of transformation and cancer clone development, if you will, we are trying to understand how they are interacting with the immune system and immune cells to be regulated, to be detected, or on the other side of the coin, to be promoted by the, if you will, chronic inflammatory cells for the development of the cancer. And in this area and this space, you can imagine that we are interested in understanding the role of innate and adaptive immune cells. We have active programs looking at these cells in conjunction with epithelial cells for this purpose of prevention. Now, one of the cell types that is quite key, at least in a textbook version of this, is natural killer cells. And these are obviously cells that everybody knows them as being innate immune cells that are naturally capable of killing tumor cells and virus-infected cells. And the work I'll be describing today and hopefully explain our discoveries on these cells really relates to their engagement in peripheral tissues. This work has been led by two really talented postdocs in the lab, Dr. Mark Bonting and Dr. Malik Vyas over the years. And the credit for the work goes to them, obviously. So just brief overview and introduction, to get everyone on the same page. NK cells and natural killer cells are basically cells, innate lymphocytes that are derived from common lymphoid progenitors. Technically, they have a distinct transcriptional factor profile to reach to the development of mature NK cells. They have one very close resembling partner in the innate lymphoid cell lineage, which are called ILC1s or innate lymphoid cell type 1s. These are cells that are, in terms of transcription factor requirement for their development from common lymphoid progenitor, have a distinct path of their development. However, as we will talk about it during this talk, they can be, if you will, developed from mature NK cells to form ILC1s, and that has been described in tumor models. The difference between what we call conventional NK cells and ILC1s has to do with the level of cytotoxicity versus inflammatory cytokine production by the cells, and more importantly, the location of these cells where they're normally found in organisms. In terms of the background on the side of mouse NK cells, so we are clear on the phenotype and the markers that we use for these cells in mice. Conventional NK cells are mostly found in blood and lymphoid organs. They do not express CD3, they express NK1.1, NKp46 in CD7 black 6 mice, and the conventional NK cells do not express CD49A, however, they express CD49B, and this distinction is what we use to mark these cells as conventional versus tissue-resident NK cells, and they have cytotoxic effector function. On the other hand, we have the tissue-resident NK cells, and that's a word I will be using in the rest of the talk. They're also called ILC1s, as I mentioned. They can be found in liver, uterus, skin, peripheral tissues for the most part, and salivary glands. And they, as I mentioned, do express CD49A, but do not express CD49B. And they're known for their inflammatory effector function within that peripheral tissue, namely expression of interferon gamma. NK cells, as everyone knows, they have a very broad repertoire of receptors that are expressed on their surface that allows them to recognize the target, either to stop the NK cells from overactivation or induce activation of NK cells. And these myriads of receptors, as you can see in this picture, is really what regulate NK cell functionality against the target. And this repertoire is obviously innate repertoire. In other words, it's not adaptive and modulated like T cells are. However, it enables the NK cells to recognize their target to a relatively good precision. This is a more restricted number of these receptors that are shown here. On the left, you can see the common activating receptors on NK cells in mice, and then on the right, you can see the inhibitory receptors. For the purpose of this talk, the receptors that are very important to pay attention to is Li49H, which is an activating receptor on the surface of NK cells in C57 black 6 mice, and also NKG2D, which is a commonly studied NK cell receptor, activating receptor, which is shared by other cytotoxic immune cells like CD8 positive T cells. And on the inhibitory side, the most important inhibitory receptor to keep in mind is Li49s of ABC, and these are basically inhibitory receptors that interact with MHC class I and prevent activation of NK cells. In a very basic, or if you will, textbook version of the description of NK cell activity, NK cells do interact with the receptors with the Li49 in mice, and KIR receptors in human NK cells interact with MHC class I on the target cells, for example, tumor cells, which in case of NK cells, this interaction leads to inhibition of NK cells. However, as you can imagine, this interaction between MHC class I and the peptide with the cytotoxic T lymphocytes leads to their activation and killing of tumor cells. And one of the primary observation that we are all familiar with is the fact that the tumor cells many times remove and lose their expression of MHC class I, and as a very prominent means of primary and acquired resistance in context of immunotherapy to evade cytotoxic T lymphocyte mediated killing of them. However, as all of us know, we are always wonder why these tumor cells were able to get away with this evasion of CTLs and not be targeted by NK cells in that context. So to be more specific, there is the concept of missing self, which is if tumor cells or a target cell loses the expression of MHC class I, this loss would result in activation in NK cells because you remove the inhibitory ligand receptor interaction leading to lysis of the target cell. However, one thing to remember is that the presence of MHC class I as an inhibitory ligand is most of the time is dominant over presence of an activating ligand receptor interaction. You can overcome this inhibition by the presence of MHC class I with increasing the level of activating ligands, what we call induced self process that then eventually can lead to the lysis of the target cell. But the concept of inhibition dominating over activation receptor ligand has been known in the NK cell field. So we started our effort now several years ago looking at NK cells, understanding this fundamental dilemma in the field of NK cell biology. And this is called hybrid resistance. And I will be explaining this phenomenon in more detail. So hopefully that would enable folks to realize what has been going on in terms of understanding how NK cells function in vivo. If you go back to textbook of immunology or go pretty much 60 years back into early and fundamental discoveries on MHC class I and MHC class I mismatch for T cell activation, this is one of the very fundamental experiments that you will see in that area, which is crossing two strains of mice and the development of F1 progeny. And these F1 progeny mice are able to accept skin graft from either of the parents. In other words, this mouse can tolerate both of the HLA type 1s of both parents' parental strains, and there is no skin rejection happens in the F1 mouse. A few years later, it became clear that when this process is repeated, but instead of using a skin transplant as a source of transplanted organ, using bone marrow in this case, the bone marrow injection into the circulation of the F1 progeny would lead to the rejection of the bone marrow. And that was called hybrid resistance. And over the following few years, it became clear that the reason for the rejection of the bone marrow cells is NK cells targeting these bone marrow cells from either of the parents in the F1 mouse. These are NK cells, of course, are licensed toward the other strains, HLA class I or MHC class I. In following few years or a couple of decades later, it was also shown that in the context of loss of MHC class I expression by removal of beta-2M, the donor mouse not having this MHC class I expression at the surface, transfer of the skin from this mouse to a syngeneic C57 black 6 mice that does have, it's wild type basically, does express MHC class I, does not lead to any skin rejection. However, if you do the same experiment again and transfer bone marrow from beta-2M deficient mice into the recipient animal, now you have a complete rejection of the bone marrow cells within the circulation by the NK cells. So this is a clear dilemma in the field, which is going back to 1960s. Why didn't the NK cells reject the skin in the first place, and how that led to all the other discoveries in MHC class I and T cells, but bone marrow did not show that, and bone marrow got completely rejected. And the question is, why are NK cells unable to kill their targets in peripheral tissues, namely in this case, skin transplants? So to address this question, we came and we tried to understand what the reason behind this dilemma has been. And to enable these studies, we have developed mice that transgenically expressed a very potent activating ligand for NK cells, and that's MO57. It's a cytomegalovirus encoded MHC class 1-like protein, which is GPI anchored to the surface of the cells. And these ligand, importantly, only binds to Li49H on NK cells. So this allows you to specifically activate NK cells without having to deal with CD8 T cell direct activation. The problem that has been really in the field of NKG2D always have been an issue with both cell types responding to the same set of ligands. Here we are dealing with a ligand receptor that is specific to NK cells. And what we did was basically repeating those very old experiments using a skin source or skin graft that expresses MO57, so activating ligand for NK cells, and also lacks MHC class 1 by the removal of beta-2M from that skin transplanted recipient animals in a syngeneic background. So when we did this experiment, we decided that these groups of mice, so wild-type MF7 alone, beta-2M deficient, or combination of the two, will be transplanted to reporter mice, which are syngeneic model, but in these reporter animals, that NK cells are green, so they're fluorescent green, and the rest of the animal cells are red. So when we did this experiment, this is basically the result we got, which is lack of rejection, even in the presence of a very strong activating ligand for NK cells, and loss of MHC class 1. We do not see any partial even rejection. If at most we see inflammation and some dermatitis at the site of the skin graft. And if this is a graft size over time, which doesn't show major changes in MF57 expressing MHC class 1 deficient grafts. However, in contrast, if we did this experiment and just use the beta-2M deficient bone marrow, like the previous publications have shown, you see a very robust rejection of the bone marrow cells that lack MHC class 1, even without adding the MF57 on top of that. So from this, we looked at the understanding first, do NK cells even traffic to the transplanted organ, in this case, the skin? And the answer is yes. These skins, especially the ones that are expressing MF57, are very much infiltrated with the massive number of NK cells into the stroma or dermal regions of the skin, as you can see here with the green signals in the dermis of these skin grafts. And what you also appreciate is that the massive induction of T cell response into these grafts as well. Again, the combination of two did not result in the rejection of the graft. So we wondered, one of the questions that came up was, if we are seeing the basically clear trafficking of the NK cells into the graft, are these NK cells becoming hyper-responsive in exposure to abundance of activating ligand, a phenomenon that has been seen in the, let's say, for example, MF57 transgenic mouse itself. So what we tried to address here was to understand if this is really hyper-responsiveness phenotype or really in situ inhibition of the NK cell killing function. So at baseline, if you take these cells out of the skin and look at the degranulation levels, they're not that different compared to the NKs from the recipient spleen. However, after 4 hours in culture, you can appreciate that these NK cells that are coming from the skin are much more active in degranulation compared to splenic NK cells, showing that they don't have any deficit in responsiveness and activation in terms of degranulation and cytotoxicity. So we wondered whether the activation in vivo within the skin graft is just not sufficient due to lack of cytokines, and we could perhaps stimulate these cells further to get them to reject the skin. So we used two very common platforms for NK cell activation in vivo, which is one of them includes poly-IC treatment plus IL-12 and 15. And the other one is a triple cytokine treatment, which is commonly used to activate NK cells, both in the context of marine studies, but also in human NK cell field as well. And when we do this type of treatments using the regimen shown here at the bottom, we still don't see any effect on rejection of the skin that expresses a ligand MF57 and doesn't have MHC class 1 expression. So then we wondered what is happening to these NK cells as they enter the skin over time. And one of the discoveries that we have made in this context is that the NK cells that are entering the skin are, over time, they're transitioning to more of a tissue-resident NK cell phenotype. So they're losing CD49B expression and gaining CD49A expression in the skin. And this has been quantified here at the bottom. And you can appreciate that in the skin that is marked by blue down here, which is MF57 beta-2M knockout, they have the highest abundance of these what we call tissue-resident NK cells. And we went on to demonstrate these are truly transitioning from conventional NK cells by using parabiosis and marking the cells that are exiting the circulation and following them over time in the skin transplant to demonstrate that this is really recruited from the circulation initially as a conventional NK cells, and then they transition to form a tissue residency phenotype within the skin over time. So based on this discovery, we wondered whether this transition is really preceding and causing the inability of the NK cells to target the skin directly and kill the skin transplants. And to address this, we relied on some of the understood literature on how these conventional NK cells can become ILC1s or tissue-resident NK cells. And one of the major cytokines that drive this transition is TGF-beta, as you can see here, and that has been described in context of tumors. So we went on to look at this effect of blocking this transition on the phenotype of NK cells and skin graft rejection. So we generated mice that are either lacking TGF-beta receptor 2 on their NK cells, or their global knockout of Hobbit, and Hobbit transcription factor has been shown previously to be important for the development of tissue-resident NK cells. So we compared these two groups of mice as recipients to a wild-type animal that is receiving the most NK activating graft that we have, which is MR57 expressing beta-2M deficient. And this transfer was successful in the sense that we were able to reduce the number of, both in the liver of the recipient mice, but also most important in the donor skin, we were able to reduce the number of tissue-resident NK cells. However, we did not see any effect on the skin graft itself. So the skin graft still, despite the fact that these mice were much less able to form tissue-resident NK cells, they did not reject the skin graft over time. So going back to an image I showed earlier and trying to look at this phenomenon of skin rejection, as I mentioned in the previous slide on this topic, that not only we see induction of NK cell recruitment to the skins, but also we observe recruitment of CD4 and CD8 T cells. And as you can appreciate, and it makes sense, in the context of beta-2M deficiency, the number of CD8 recruitments are much less than what we see in MR57 alone expressing skin grafts. And that brought us to our attention that perhaps NK cells are coming to the skin, they're activated, but they're activated as an inflammatory cell, more of an helper cell, and really require the T cells to be present for the rejection of the skin. And by using this model of activating NK cells, we are really losing the efficacy of CD8 positive T cells. So to address this and try to understand that, can we really get this rejection phenomenon to work, even though the NK cells we are understanding are not the direct killers, but are they activating other T cells or other immune cells to do the effector function? We moved to a F1 transplant system in which you could maintain the functionality of CD8s by maintaining the MHC class I expression on the graft, but also give the NK cells the full activation because they're looking at the graft as a missing self. In other words, to them, the ones that are licensed against the other MHC class I, the graft would be a missing self type of target, so they would appreciate that as a lack of MHC class I. So we generated mice that are F1 breeds of C57 Black 6 and BALB/C mice, and then we transplanted MR57 skin, in this case, and beta-2M competent skin grafts on back of these F1 progeny, and we understand that these skin now can be targeted both by NK cells at the full manner of NK cell activity, and then also be seen by CD8 positive T cells. So when we did this experiment, it was a clear rejection phenotype was observed in these mice. These are acute rejection of the skin after 20 days. And what was very interesting and confirmatory for us was that even though this rejection, again, the ligands that enables this rejection, MR57, is specific to NK cells, the rejection phenotype pursued what we call adaptive clock. In other words, the rejection starts happening after day 12 after transplantation. And this is somehow very late for an innate rejection phenotype, but it very much follows how CD8 T cells would target and kill a transplanted skin on mice. If you look at the infiltration of the immune cells into the grafts that is being rejected, you can see that the NK cells are coming in around 5 days after transplant into the graft at high numbers, and they accumulate over time. Again, if this transplant rejection was a direct function of NK cells, you would expect them to show you phenotypes before day 10 because the numbers and the efficacy would be there to reject directly. However, the rejection phenotype follows the accumulation of T cells, and the T cells are entering the skin following the NK cells. So it provides a nice temporal evidence that NK cells are really recruiting, functioning as helper cells to bring the T cells into the skin graft, which then eventually leads to the rejection. We wondered if this is required, meaning are these NK cells and T cells required for this rejection? And we demonstrated this using elaborate methods of depleting these cells. As you can imagine, we were trying to deplete these in the peripheral tissue, and we had to use both DTR systems on top of antibody treatment to achieve a complete depletion of the immune cell target. And then we did that in both cases, actually, both cases of removing NK cells by using mice that are expressing DTR within their NK cells, treating with diphtherotoxin and anti-NK1.1 antibody, or removing all T cells by using CD4 ROSA DTR and diphtherotoxin treatment plus anti-CD4-CD8 antibodies. We can reverse the rejection that you see in the control animals up here. So this phenomenon of rejection is both NK cell and T cell dependent, again pointing back to the concept that the NK cells are coming to the skin. They're activated, but their activity is different than what we are thinking of NK cells as cytotoxic agents. And they're really providing that inflammatory or helper function to the other lymphocytes for them to function as effector cells for the rejection. So then we really wanted to understand what is happening to these NK cells. So this is, again, the reminiscence of many of the phenotypes others have seen in tumor models and many other viral infection otherwise. So we had a window here, which was a very clean window, if you will, because you're dealing with a normal skin transplant that allows you to really time this out and try to understand what happened to the NK cells as they enter the skin. So that is exactly what we did. We took NK cells from the spleen of the recipient mice, from their blood, from the donor skin and recipient skin, and profiled them to understand what is really going on with the NK cells as they're encountering the skin graft. One of the gating strategies, which is paramount to understanding our finding, is this gating here on the donor skin. So what we are trying to do, we are trying to separate the conventional NK cells from tissue-resident NK cells within the donor skin. And both of these cell populations that we are sorting and sequencing are coming from cells that are outside of the circulation. In other words, when I say it's a donor skin-derived conventional NK cell, it's an NK cell that is already outside of circulation because it is not marked by CD45 IV antibody staining. So it's really has the cell that has exited the circulation is in the skin microenvironment, but is still is at the stage of being conventional NK cell. In other words, they're immediately exiting, the ones that just exited the blood into the skin. And if you look at the profile of these cells, it's quite striking. And this is really probably the most important finding here, which is we were expecting that some of these transitions of NK cells would happen at the time of their transition to tissue residency. However, many of these changes from cytotoxicity to inflammatory phenotype happens right at the point of their exit from the circulation. In other words, this data suggests that this transition is not necessarily delayed epigenetic modification of NK cells over time, but it's really an immediate signaling effect that is modifying the function of NK cells from killers to helpers. And if you look specifically at these NK cells, these are, again, conventional NK cells. We're comparing the ones that exited the blood versus those in circulation. Same idea. Again, you have increase in chemokine, cytokine expression by these NK cells and loss of several of these cytotoxicity-related genes. And we also have induction of TIGIT and some of the other immune checkpoint molecules on the surface of these NK cells as well. This is gene set enrichment analysis nicely demonstrates that concept I mentioned, which is loss of NK cell mediated cytotoxicity and gain of inflammatory response as the NK cells exit the circulation and as soon as they enter the skin. And that's really the key concept to take away from this analysis. We then wanted to understand, can we find markers that would define these conventional NK cells that are now really not like circulating conventionals, but they're conventionals in the peripheral tissues. And as you can see, as I mentioned, TIGIT is somewhat expressed, or some other receptors that we see some inductions in. But one of the receptors that was quite interesting, quite telling actually, is the clear induction of TGF-beta receptor 2 on these NK cells. In other words, it makes, if you sit back and think about it, it makes perfect sense. So the NK cells are coming out of the blood, they are upregulating TGF-beta receptor 2 and eventually becoming susceptible to TGF-beta signaling for their eventual transition to tissue residency. But their phenotype change that prevents them from direct cytotoxicity well precedes the transition to the tissue residency phenotype. And that's really the key take home here. And we were obviously saw those TIGITs and other PD-1 and some of the other checkpoint molecules on the surface of these NK cells as they exit. So we wondered, can we block those and perhaps get these NK cells to stay cytotoxic and provide skin graft rejection? And the answer is no, you can't really modify their phenotype with blocking any of these or combination of these checkpoint molecules on the surface of the NK cells. So then we went back basically to drawing board. So we have many NK cells, as you can see in this picture, coming into the skin. This is day 10 after MF7 skin graft placement on the mouse, on the reporter mouse. So the skin is loaded with NK cells, and they're not doing their job of direct killing of this target, which is basically the best target you could provide to these NK cells. Again, if this target was provided in the blood, these cells would be dead, no question, in 72 hours max. So we wondered, what are the NK cells now coming in contact with, because we are now understand that this is more of a signaling interaction and very immediate effect on the NK cells that result in this effect. So if we go back to the earliest time point that we saw the NK cells entering the skin, and that was day 5 post-transplant, you can appreciate that majority of these green cells are sitting either embedded into extracellular matrix, or they're in rare cases contacting fibroblasts that are coming with that skin transplant on the back of the mouse. So we basically ask a very simple question: are these elements that the NK cells are coming in contact with on that early time point within that skin graft, are the ones that are modifying NK cell functionality? So first, we went after the fibroblasts. So if you take the conventional NK cells from spleen and expose them to embryonic fibroblasts that either express nothing or express the MF57, MF57 expression would lead to a major activation of these NK cells. We don't really see an inhibitory effect from the fibroblasts on NK cells, as shown here. On the other hand, and that was obviously a surprise to us, when we modified this protocol to look at the role of the ECM proteins in this interaction using this platform, which is taking the NK cells, conventional NK cells from spleen of the reporter mice, having them co-culture with the ECM proteins for 2 hours, and then exposing them to the MF57 expressing embryonic fibroblasts, we saw a clear phenotype of inhibition by collagen 1 in this case, and also elastin in this case as well. We went on to look at some of the other effects that we have seen previously, and one of the ones that was at this point very telling was this data I showed you previously. In this data, as I mentioned, we demonstrated that if you take the NK cells out of the skin graft at baseline, they do not show any cytotoxicity or degranulation, CD107A expression. However, after 4 hours in culture, again, these are single cell preps of the skin graft that has NK cells in them. NK cells now fully activated, and this 4-hour single cell culture is basically done after collagenase treatment. So you have to remove the collagen and extracellular matrix protein to enable this culture system and interaction of the NK cells with the other skin cells that are present in that skin prep. And we believe that collagenase treatment is really what allowed for NK cells to be reactivated in this context. On the other side, I mentioned the inhibitory effect of collagen and elastin on the NK cells in terms of cytotoxicity, but these same collagen 1, and in this case collagen 3 as well, induce expression of chemokines that we saw in our RNA sequencing from the skin, from the NK cells that we got from the skin graft. In culture, you can activate the expression of these chemokines by the conventional NK cells that are coming from the spleen. Among the possible interactions and signaling pathways that could regulate what we are observing is LAIR1. LAIR1 receptor has been described as an inhibitory receptor that is expressed by immune cells, including NK cells, and is able to inhibit cytotoxicity and inhibits T cell function for the most part has been studied. So we wondered, is this LAIR1 collagen interaction is the main driver of this inhibitory effect against the cytotoxicity of NK cells? The answer is somewhat. So in other words, if you use LAIR1 knockout NK cells in culture, and in the absence or presence of collagen 1, you do get partial reversal of that inhibition of cytotoxicity and interferon gamma expression. However, this reversal is not complete. So what is going on? As you can see here, NK cells express a multitude of receptors that could bind and interact with ECM proteins. In fact, the two markers that many of us in mouse NK fields use, CD49B that I mentioned to you as a marker for conventional NK cells, and CD49A, which is a marker for tissue-resident NK cells, are integrin receptors, basically. So these are, their functionality is not really studied well, but they're present on these NK cells, are modulated, as you can see, as they exit the circulation, coming into the skin. And there are multiple of those, which we are currently obviously studying right now, and as this field is opening up and trying to understand what these receptor ECM protein interactions mean in the context of NK cell activity. So we wanted to, at least for the first try, we wanted to understand, can reduction in ECM proteins improve NK cell cytotoxicity in peripheral tissues? And we unfortunately could not do this in the context of a skin transplantation, because if you remove ECM in a skin transplant system, you basically destined to graft failure. You can't really have the graft taken in mouse. So for this reason, we had to switch over to a cancer model in which we could modulate ECM production and still have a tumor to study for their NK cell effects against them. So we went to a very classic tumor model used in NK cell field for immunological analysis, and that's B16 melanoma models. Beta-2M knockout B16 IV injection is basically a classic model now to look at metastatic melanoma and the role of NK cell in it. It's well established that if you remove NK cells from mice using antibody depletion, the mice that are injected IV with beta-2M B16 will get a lot more tumor foci in their lungs compared to the control-treated, IgG-treated animals. But if you take that same beta-2M knockout B16 cells and inject them subcutaneously, you don't see a difference in terms of presence or absence of NK cells. It confirmed, at least validated what we were hypothesizing in context of skin transplant tumors as well. So NK cells are again marked here by reporter GFP. They're coming around the tumor foci. This is 3 days after implantation of the tumor, and they're almost embedded in the collagen matrix that is laid out around this tumor foci in the mouse. However, as you would expect, the NK cells do control the metastatic, if we will, phenotype of the subcutaneously injected melanoma cells, and you can see more lymph node presence of the melanoma cells in mice that received or got depleted for NK cells compared to the IgG control. So we went on to try to reduce the content of collagen and ECM proteins in these tumor models, subcutaneous melanoma models. And we used two platforms to do that. One was losartan, which is obviously a very commonly used antihypertensive medication that can block collagen synthesis through blocking TGF-beta. And the other one was DHB, which inhibits the assembly of collagen. It's a distinct mechanism, and we use both of these agents with the understanding that both of these, using, again, different mechanism of blocking synthesis or assembly, reduce the mature collagen generation in the tumor cells and around the tumor cells, if you will, in mice. So this is the platform we used, the same subcutaneous injection of beta-2M knockout FB16 melanoma cells, and then treating them with losartan or DHB over time and trying to understand, is the NK cells playing any role by depleting them in these models. So if you treat mice with losartan, half of them get depletion of NK cells. These mice develop much larger tumor compared to mice that are competent in NK cell compartment. Same is true with the DHB-treated mice. Again, the NK cells now are showing you functionality for tumor control, because when you remove them, the tumor sizes are much bigger. And we wondered what's happening in situ within the tumors. And the answer is pretty clear. When you treat the mice with losartan DHB, the tumor cells are much more capable of entering into the tumor parenchyma compared to the PBS-treated mice. And this is shown here, quantified here. And as importantly, we also see increase in cytotoxicity phenotype and expression of granzyme in the losartan DHB-treated NK cells in the tumor compared to controls. And we also see more gamma expression and more PD-1, perhaps here as a marker for NK cell activation. So finally, we wonder, does the distinct function of NK cells in circulation versus peripheral tissue translate to humans? The short answer is yes, and that's something that we're obviously actively exploring right now, taking these fundamental discoveries from mouse into human. But I do want to close by mentioning one of a piece of data that I'm sure everybody's familiar with, but we try to put it together as one unit here, which is the understanding that all of us have, that solid cancers many times use the loss of MHC class 1 and mutation in beta-2M as a means to evade immune system, namely CD8 positive cytotoxic T lymphocytes. However, this type of selection of beta-2M alteration is not found really in leukemias, and this is not at all a predominant way of resistance in these cancers. And that suggests again that the evasion of the immune response within the circulation is really impossible if in case of by the loss of beta-2M, because the tumor cells would then become prime targets for NK cell killing. But in the solid cancers, the tumor cells are capable of doing this process because they're not really directly being targeted by NK cells, as the NK cells are losing their cytotoxicity. And again, their functionality is modified from cytotoxicity perhaps to an inflammatory phenotype. So in conclusion, NK cell functionality upon entering a solid organ like skin is immediately altered from cytotoxicity, a killer phenotype, to an inflammatory helper one. This switch precedes the conversion of conventional NK cells to tissue-resident NK cells within that peripheral tissue. NK cells play an important role in clearing circulating tumor cells, while solid cancer cells are resistant to direct NK cell cytotoxicity. ECM proteins in solid organs and solid cancers play a critical role in blocking NK cell cytotoxicity while promoting its inflammatory function. And this is, we believe, it's truly a physiological phenomenon that is happening here, one that is evolutionary, perhaps makes perfect sense, and that the switch in NK cell functionality from killer to helper in peripheral tissues may highlight the benefit to prevent an overactive cytotoxic response from an innate immune system. For example, in case of localized viral infection in the peripheral tissue, while promoting development of a more precise adaptive immunity over time. And as you can tell, this process is not favorable in case of viremia or a virus or infection that is affecting the blood and circulation. And in that context, NK cells are required to attain their cytotoxicity directly and eliminate that threat, if you will, in a more direct manner. And finally, as you can imagine, there are further investigations of NK cell ECM protein interactome is required really to understand its therapeutic potential for cancer, infectious diseases, autoimmunity, inflammatory disease, and transplantation. And we'd like to close by providing these conceptual models. So hopefully that it helps to understand this concept better, which is we have NK cells in the circulation, and these are cytotoxic, perfectly able to kill their targets directly. As they enter the peripheral tissue, in this case skin, they do have receptors that allow them to interact with various ECM proteins, and these interactions lead to the induction of inflammatory gene sets in these cells and suppression of the cytotoxicity program within the cells. And this process, as I was demonstrating in the context of a skin transplant, does end up recruiting and activating T cells against that target within that peripheral tissue. However, the NK cells over time do transition at least portion of them to a tissue residency phenotype within that peripheral tissue. This same process, we believe, is happening in context of tumors, and as the NK cells are exiting the blood vessels coming into the tumor microenvironment, they're interacting prominently with these ECM proteins that block their cytotoxicity, and some of that could be reversed by removing or modulating or reducing those ECM contents or ECM protein contents within that tumor microenvironment to enable, again, the cytotoxicity from NK cells. But eventually, some of the areas we are very interested in pursuing is modulating the receptors, identifying the receptors or prominent receptors that are really important for this transition and this modulation, and trying to modify them and enable NK cells for better efficacy depending on the disease of target that we are trying to address. And with that, I would like to thank the member of the laboratory who did the work that I presented here. I mentioned Dr. Bunting and Vyas and other folks in the lab, and also our collaborator, Mike Lawrence and Rob Manguso at MGH Cancer Center. And with that, I will be happy to take questions. Thank you so much.
Thanks very much, Sean. Fantastic talk. Just a reminder, anybody who has a question, please do not type it into the chat, but rather into the Q&A tab at the bottom. And there are a number of these questions already, so we'll just dig right in. Sean, I can read them if you like, or you can see them. The first is from Jonathan Frieden: is the PD-1, PD-L1 pathway irrelevant to NK cell behavior?
That's a great question. In the models that we tested, which is a skin transplantation model where we tested the efficacy of these checkpoint inhibitors, we could not see any change in both NK cell phenotype and obviously functionality, which I showed, which is the rejection of the skin. In context of tumor models, it's still debatable. There's a debate on how effective these checkpoint inhibitors are on the NK cell functionality, but we haven't really tested those in our melanoma models.
Great. Next question from Dan Liang. Great talk. I might have missed it. Sorry. But is the hyporesponsiveness or tolerance from NK cells only observed with MHC1 deficient targets or also with MHC1 mismatch targets?
So the question of tolerance, so that's not how I would word it in the context of a skin transplant. I would say lack of, if you will, cytotoxicity. We see that in both contexts. In other words, as I showed, if you go into the mismatch context, which is the F1, and provide that missing self access to the NK cells for their full activation, we don't still see rejection as long as T cells are removed from the picture. So if you deplete the T cells and only are dealing with the NK cells within that microenvironment, you don't see any rejection. So that tells us that even in the context of mismatch and missing self for the NK cells, they're not really providing that cytotoxicity.
Next question from Amin Ali. Very interesting talk. I was wondering if you see the phenomena of the switch of the conventional NK cell from cytotoxicity to inflammatory phenotype in other tissues besides skin, especially other solid tumors. And then a second question, are markers of ITGA1 and 2 good gene expression markers to differentiate tissue resident versus conventional NK cells in single cell RNA-seq?
Those are great questions. So in terms of the other tissues, the short answer is yes, but we are still working, trying to understand those areas better. But what we're seeing is that the tumors that are in basically solid organs of epithelial types, they do have the similar phenotype of switch that happens in them. And this again has been also looked at previously by other groups in context of, by switch I mean transition to the tissue residency, if you will, in other tumor models as well. But in terms of switch in conventional phenotype, we do see them besides the skin in other epithelial organs. The markers of those two markers, again, those are used mostly as surface receptor markers for identification of conventional versus tissue residency phenotypes. I would argue at the level of single cell, perhaps a more combined expression of other genes would be required, perhaps to call them conventional versus tissue resident. But in terms of protein expression on the surface, those are established markers in mice for distinguish.
Another question from Jonathan Frieden. Do you have an in vitro model for human exploration?
That is exactly what we are working on. That's definitely we have that, and it's actually very much phenocopies what we're seeing in mice. It's quite amazing.
From Bogang Wu, thanks for a great talk. Did you check other collagen receptors in the tumor NK crosstalk, like integrin, DDR1, 2, etc.? Thanks.
Yeah, that is a fantastic question. That is basically the opening of the field. So we start with layer 1. I just was able to show you that layer 1 is partially responsible, not completely. And we are really diving into it. We are looking at integrins, DDR1, DDR2, and others actually in this context, and more to come. So I don't have any updates on those, but definitely we believe they have an effect, but we don't know which ones and the combo of what they're interacting with, because it's diverse on the side of the receptor, but as you can imagine, it's also diverse on the side of the ligand, the ECM proteins. It's not all about collagen 1, not even close. So all the other collagens and elastin and fibronectin, vitronectin, all those things seem to have a very active interaction with the NK cells in terms of signaling modulation. But that's a great question.
Next question. CD49A positive tissue NK cells was reported to be correlated with adaptive NK memory. Did you compare the adaptive features of the conventional NK versus tissue NK in your study?
That's a fantastic question. We would love to do that. We haven't done it. And again, most of that data that we have is coming from this context of skin transplantation. We haven't pursued that, but that's one of the areas that we definitely would love to look at. Yes.
Next question is from Kai Wucherpfennig. Does the inflammatory NK cell population in tissue express XCL1 and CCL5 and thus recruit conventional DC1?
XCL1, I don't know on top of my, I can get back to you on that one. CCL5 is actually downregulated in NK cells as they enter the skin, and that specific chemokine is regulated by ECM. So we have pretty decent data on that as well, that the conventional NK cells from spleen exposed to, I believe, both collagen 1 and collagen 3 immediately downregulate CCL5. And that's something that we're observing in this NK cells.
Next question from Bart Luderbach. Are B16 beta-2M null primary tumors better eliminated in Lair null mice?
That's a great question. We have looked at that. The answer is not conclusive. We currently have, but we didn't have it when we were doing the experiments, and mice that lack LAIR1 specifically on NK cells. We did have the LAIR1 global knockout, and we could not discern the change. And that's, again, one other reason that we believe there are other receptor ligand interaction we should pursue, but we're also making the mice that are LAIR1 deficient specifically on NK cells to go back at that question.
Question from Harmony Saunders. Great presentation. I also love the summary graphic. Have you noticed changes in any other collagens aside from collagen 1 that may be suppressive?
Yes, as I showed, in terms of collagens, depends on what axis we're going after. In terms of the cytotoxicity, collagen 1 seems to be the dominant one in the collagen side. Now, elastin has a very strong effect as well. Collagen 4, I believe, didn't have effect at 9 hours, but over longer incubation also has an inhibitory effect, I believe. We haven't tested, you can imagine, we haven't tested many of the other collagens, mostly the main ones we found in the skin, or you can find in the skin. So yes, I would say maybe 2 of them had the inhibitory effect, but not all of them. And yeah, and then there are some that actually have some very different effects on NK cells as well.
Question from Thomas Tan. Has anyone implicated tissue-resident NK cells in skin-related adverse events due to their heightened pro-inflammatory response?
Not that I know of, and that's one of the things that we were able to demonstrate this transition in this model, because meaning the actual transition enabled the understanding that in this skin transplant model was really because skin at baseline doesn't have many NK cells in it. So I'm not aware of the tissue-resident NK cells being highly implicated in the broadly speaking, in inflammatory skin diseases for that matter, because it's not a big population within the skin. Yeah, we see them again forming. We spend a lot of time looking at that question. Are we recruiting tissue-resident NK cells from other sites into the graft, or are we transitioning a conventional NK cell into a tissue residency? And the latter is correct. And one of the ways we were able to do this was because the surrounding the skin doesn't really have much NK cells to start with.
Question from Takashi Nishina. Does TGF-beta signaling directly affect NK cell function because the proform of TGF-beta is also enriched in a collagen-rich environment?
Our understanding is it's basically a mature TGF-beta signaling, cytokine signaling to NK cells that is really helping the transition of these conventional NK cells to tissue residency. We haven't really looked at, that's an interesting concept. We haven't looked at it in the context of proform of TGF-beta and its effect. Yeah, but that's definitely something we could definitely look at.
A couple more questions, time permitting. Harmony Saunders. Also, if there's time, you saw that the fibroblasts are not suppressive, but the collagen might be. Do you have a speculation on a potential mechanism?
Yes. So I didn't have a chance to show that here, but we do believe it's the collagen, or I would make it broader, ECM protein ligand as a ligand, and receptors expressed on NK cells that modulates their function. It actually is signaling modulation, so you can look at the downstream molecules within the NK cells, and we have done that. And when you expose them to these various collagens, you do see that signaling modulation almost immediately, within 2 to 5 minutes in some cases, of changes within the NK cells that are happening in exposure to these ECM proteins.
Another question from Jonathan Frieden. How long do you imagine it might take to get to a therapeutic trial? And what form do you imagine such a trial might take?
That's a very, very important question. That is one of the other reasons that brought us to look at human side of things, and we are seeing the same phenotypes, which is how we would envision utilizing this therapeutically. And one of the areas obviously could be like looking at it as an antibody blocking of the receptors, and that may not have NK cell specific effect, but looking at it more broadly as receptors that I would also argue are important in T cell biology as well, again, beyond layer 1, looking much more broadly as the ECM repertoire with the immune cells. And that's something that our lab has taken on to explore. But more specifically is the question of the cellular therapies and NK cells, both adaptive NK cell therapies and also CAR NK cells. And in that context, we are trying to, in collaboration with folks here, both Dr. Marcelo Mas in our institution, as also Rezvan over at the Dana Farber, trying to understand and try to get into engineering some of these receptors in ways that we are learning about them and in their interaction with ECM proteins. So I would say it's actually very proximal to therapeutics, at least conceptually, in order to be able to testing them in reagents that could be potentially used in humans.
And related to that, Venkateshwar Keshamuni asks, great talk. Do you think even CAR NK cells encounter the same problem of getting converted into tissue-resident NK cells?
I don't know about converting to tissue-resident NK cells. That data we don't have. But as I presented here, we believe that the switch in loss of cytotoxicity of NK cells is a very immediate effect in NK cells. And if that's the question, which is, do we see the switch in, could the same switch or removal of cytotoxicity from NK cells happen to CAR NK cells? The short answer is yes.
And I just have one last question. Could you, since losartan is a pretty common human antihypertensive, could you look at patients on, let's say, melanoma patients on immune checkpoint blockade who are on losartan and see if the accumulation of MHC low or beta 2 deficient is different in that population?
That's a great question. We haven't looked at it specifically. One of the reasons we quickly, from these initial studies, are moving into antibody-based or modulation of the receptor is, as you know, myriad of functions from losartan. There's some obviously very amazing data from Rakesh Jain's group looking at losartan and immunotherapy in pancreatic cancer, and they're seeing efficacy in that context. But again, as you know, there's a TGF-beta effect directly. There's multiple effects that this drug has. We could technically look at that. So definitely something we could explore and see if we see any signal specifically as it relates to NK cell functionality. But again, I would also argue, because it's really modulating collagen, you might also see T cell functionality, that some of it might be related to the modulation of collagen, not necessarily just TGF-beta directly.
Yeah. Last question from Jonathan Frieden. Can you extrapolate from your experiments with mice what side effects blocking some of these receptors might cause?
Yes, that is really the core of this discovery, which is we are not really showing here how NK cells are modified pathologically. We are showing here how NK cells are regulated physiologically, and that's really the core of this discovery. That's why we actually started with the skin transplantation, because we really wanted to stay away from a tumor model and look at this concept in a more physiological way that is much simpler to dissect. And that's what we're finding, which means in the context of, as I mentioned, if you're starting to modulate these NK cells broadly and not targeted in the sense of tumors and other things, we might be dealing with, changing something that was evolutionary favorable for human or for the host. And that's cytotoxic activation of these innate cells in periphery, which could, again, lead to chronic inflammation, could lead to, again, off-target effects or off-target cytotoxicity from NK cells. So that's really something that we are trying to gauge and understand how this modification, how much of the modification needs to happen on NK cell side versus on the ECM protein side locally in that microenvironment? But that's really fantastic question. Thank you.
Okay, well, that was a marathon. Fantastic. Great questions from so many people. Fantastic lecture, Sean. And
thank you,
everybody. I think with that, we've reached the top of the hour, and we'll end the session. Thanks very much, everybody.
Thank you so much.
Bye-bye.
Thanks, everybody.